My name is Brett Trost, and I'm a research associate in Dr. Steve Sheridan's lab at the Center for Applied Genomics at the Hospital for Sick Children in Toronto. And of course, the Center for Applied Genomics is one of the three CGEn nodes in Canada. And before I kind of get into who I am, I just want to say kind of what a privilege it is to work here and kind of what a nice thing it is to be able to work at one of these CGEn nodes, just because you're really close to where the data is being generated and you really feel immersed in everything that's going on and the new technologies that are being developed and used. So my research primarily focuses right now on human genetics, but that's not actually my background. So I did my undergrad degree, which was a very long time ago now, at the University of Saskatchewan. And the title of my degree was bioinformatics, which in practice was a combination of mainly computer science, but also a bunch of life science related subjects like biology, biochem, other life science related subjects. And at the time, I think that was quite an unusual degree. So I think the University of Saskatchewan was one of the first universities in Canada and maybe even in the world to offer a degree that's dedicated like kind of directly to bioinformatics. But I really enjoyed it. And I think it was sort of set me up very well for a research career that focuses on the application of computer science to biological problems. And then I ended up doing both my master's and my PhD degrees in computer science at the same university, University of Saskatchewan. And those degrees also focused on the application of computer science to learning more about biology of health and human disease. But it wasn't actually until I joined Steve's lab at SickKids that I started really focusing on human genetics. And my specific focus in Steve's lab has been on whole genome sequencing data, the analysis and interpretation of whole genome sequencing data. So the beauty of whole genome sequencing data is that it theoretically allows you to detect all sizes and all types of genetic variation in just a single experiment. So previous technologies kind of let you detect some types of variants, but not others. But whole genome sequencing theoretically allows you to detect everything. And that makes it really powerful because many diseases have very complex genetics, and it's really important to kind of be able to look at all these different types of genetic variants. And I'll talk about that a little bit later, I think, as well. So for the first few years in Steve's lab, I mainly focused on kind of technology development or looking at how we can kind of better use whole genome sequencing data in order to detect different kinds of genetic variants, especially copy number variants, which Steve was one of the pioneers in discovering copy number variants to begin with. But more recently, I've been focusing on autism specifically, whole genome sequencing data in autism, which is a condition that's very complex from a genetics perspective. And much of this work in autism has involved developing, analyzing data from a resource called Missing, which is actually the world's largest data set of whole genome sequencing data in autism. So yeah, I think that pretty much summarizes my kind of work recently and a long time ago.
Amazing, great. Thank you for sharing that. It's very interesting to hear your background, and it's great to hear why you're so excited about the work that you're doing now. So can you tell us what's something new or surprising that you've learned lately in your work?
Yeah, so I'm actually not totally sure if my answers are going to be strictly new and surprising, but maybe more about sort of the lessons that we learned or that come out of the recent study that we published on autism whole genome sequencing. So I think the first big message that comes out of that paper is how important it is to look at all types of genetic variation. So when you're like some diseases are kind of simple, or at least simpler than a disease like autism, is that there's just one gene or maybe a few genes that are involved in that disease. But autism is kind of different in that there's many different genes and many different types of variants that are involved. And it's really impossible to get a comprehensive sense of how genetics contributes to the susceptibility of autism unless you look at all different types of genetic variants. And that's really what we tried to do in this study. So where many prior autism studies would just look at one type of genetic variant at a time, we tried to look at everything in one study. So in terms of sort of how the different types of variants that we looked at can be classified. So one way of classifying them is by size. So we looked at everything from single base changes, which is like the smallest possible change that can be made to DNA, to really large genetic changes that can affect entire chromosomes. And another sort of way in which we analyzed the data was based on the frequency of the variant in the population. So in autism, most of the variants that we know are associated with autism are really rare in the population, which makes sense because it's not, a really common disease. So most of the variants that we find, they're rare and they damage a specific gene, and that's how we can tell that they might be associated with autism. But then there are also variants that are very common in the population and which contribute just a little bit to someone's susceptibility of getting autism. But if you add up the effect of many thousands or tens of thousands or even hundreds of thousands of these common variants, each having a very small effect, when you add up the small effects, you actually can get a fairly large effect. And that's another thing that we analyzed in our study. And then a third way of categorizing these variants is where they actually occur in the genome. So most genes and most autism-associated genes and variants are found in our nuclear genome, which is where most of our genetic material is found. But our mitochondria, which are sort of the energy powerhouses of the cell, also have their own genome. And we analyzed the mitochondrial genome in our study as well and found some autism-associated genetic variants. And I think that we also looked at sort of other categories of genetic variants like canon repeat expansions, which is an interesting type of variant that we actually published a paper solely on a couple of years ago, and also variants in non-coding regions of the genome, which are quite difficult to interpret, but we tried to look at those in this study. So I think that the lesson there is that, yeah, like if you look at all these different types of genetic variants, you can find things that are associated with autism or whatever disorder you happen to be looking at. But you really need to have whole genome sequencing data in order to do this, because it's the technology that allows you to look at all different types of genetic variants in just one experiment. And then I think the second lesson from the study is kind of more specific to autism, but it also would apply to other diseases. So in autism, there's been lots of studies on the heritability of autism. So the heritability is basically like what proportion of autism can be attributed to genetic factors. And twin studies have been a big source of the analysis of heritability in autism. And twin studies generally show that autism is about 60% to 90% heritable. But the thing is, even in our recent study where we looked at all these different types of genetic variants in autism, we were only able to find explanatory rare genetic variants in about 15% of the autistic kids. So there's this really big gap. So 15% versus the 60 to 90% that we think is attributable to genetics. So what kind of is going to fill that gap? So I think this kind of highlights how, even though it's been, over 20 years since we first sequenced the human genome, it still kind of highlights how immature our understanding of the genome is, and we really need to, learn a lot more about how the genome is organized and how it's regulated in order to kind of be able to fill in that gap between the 15% and 60 to 90% and better understand the genetics of autism, as well as other conditions that are genetic in origin.
Excuse me, that's amazing. In terms of like the scale and the scope of the work that went into the study, can you talk a little bit about that? Like, what is the importance of, the collaboration that's involved with the missing project, and in teasing apart all these different findings, like where do you go next with this, and how much does collaboration play a role in that?
Yeah, that's a great question. I mean, collaboration played a huge role in this study. So the missing resource that I mentioned earlier is a collaboration between mainly SickKids and a company called DNA Stack, which does a lot of the technology work for missing, Google and Verily Life Sciences, who also contribute to the technology aspect, as well as Autism Speaks, which we work with to recruit participants and which funds a lot of the work. Another kind of way of looking at the collaborative nature and interdisciplinary nature of the work is just to look at the list of authors in the Cell paper that we recently published. So I think the count of authors is over 100. And one thing I really like to point out actually is that if you take that list of about 100 authors, and let's say you just pick any two authors at random, A and B, like the chance that author A would have been able to do what author B did for the paper or the other way around is very small. So like the range of expertises that were necessary in order to make this paper happen is just mind-blowing in a way. I mean, I wouldn't have been able to do what 99% or 95% of the other authors did. So, I mean, everybody's contribution to that paper was, totally indispensable. And just the range of expertise that was necessary is just amazing.
Amazing. So I have two follow-up questions to that. So the first is I would love to learn more about what your specific role was in the work and sharing your experience about that. And then what are the next steps and how do you kind of coordinate those next steps after? Because there's obviously more work to be done. So what does that look like?
Yeah, so my role is mainly on the computational side because that's my background. So a lot of the sort of computer science specific things I was involved with. But also just in terms of organizing all the collaborators. I mean, obviously Steve played a very, very important role, probably much more than I did in terms of sort of getting together all the people necessary in order to make this happen. But basically once Steve assembled the team, my role was, at least in part, to try to kind of organize everything and make sure that all the different parts of the project were on track. So in terms of next steps, I mean, I think even though we looked at all these different types of genetic variants in autism, I mean, there's still many different things that we didn't look at. So, for example, we didn't look at somatic variants, which are variants that are not in the germline, but occur, after someone is conceived. So these also play a role in autism, but they're kind of difficult to detect using current technologies. It can be done, but it's just quite difficult. So that's one thing that we didn't look at in this study. And another kind of genetic aspect that we didn't look at in this study is epigenetics. So these are kind of changes made to DNA, like methylation, that affect how genes are regulated. But the usual short read sequencing technology that we use for this study doesn't detect methylation. So we have some data from other technologies, but not for the whole data set. So for that reason, we didn't really look at that in this study. And there's also just other kind of types of variants that we didn't look at in this study. So even though we kind of call it comprehensive, it's not sort of truly comprehensive in that we looked at absolutely everything. There's still a lot of things to look at. And going back to sort of my previous point about like how, like, sort of our immature understanding of the genome. So there's a few different hypotheses that I kind of have in mind to look at in autism in terms of how can we use information that we know about the genome in order to sort of better understand genetic variation in autism. And these are the kinds of things that I'm working on right now.
Great, thanks so much for sharing that. So in terms of what do you think other people who are using genomics, other researchers or other users should know about in terms of genomics? And there's obviously applications in pretty much every field right now that we're seeing. So, but you as an expert, what would you recommend people to be aware of in terms of how to use genomics for their research?
Yeah, I mean, I think that goes back to the first lesson that came out of the paper is that it's really important to look at all different types of genetic variation. So, I mean, often people are, they'll have their pipeline set up for, say, looking at small variants, or they'll have a pipeline set up for looking at copy number variants. And because they sort of have existing pipelines or expertise in looking at those types of variants, that's all that they will look at. But, if you only look at specific things, you're going to be missing things, or at least for most diseases or conditions, you're going to be missing things. So I think the importance of looking at all different types of genetic variation is something that needs to be highlighted.
Great. And I also noted that I think families were pretty involved in this study. So in terms of what, like the public should be aware of, or even participants in these types of studies, I think you're kind of alluding to the fact that there is so much unknown. So in terms of what their expectations are when they participate in these types of studies, what can you share about what that process looked like for the researchers and how they worked with the families and the children who were participating?
Yeah, that's a great question. So the participation of the families is really important in the research study in missing. So you know, one thing I think that's really important is that their participation doesn't necessarily stop after they sign the consent forms and give their samples in order to participate in the study. So we regularly interact with these families and solicit their input on how the study should go, and whether they have any feedback on what we're focusing on. Another thing that we do is, if we find like a clinically significant variant in a given patient, we'll return those results to the patients. So I think I'm not sure about the exact number, but like hundreds of families in missing have received genetic answers for why their child has autism. And I think this is very important to some of these families, in that, like, a lot of the kind of difficulty that they struggle with is just the unknown, like, why does my child have autism? And the fact that some of these families can actually receive genetic answers and get some clarity and understanding into what's going on really provides sort of a level of comfort in getting that understanding of why things are the way they are. And actually, another thing that I'll mention is that we're involved in another project called the Autism Sharing Initiative. So what the Autism Sharing Initiative is a collaborative effort to try to combine different autism data sets from around the world. So in the initial sort of release of the Autism Sharing Initiative, there'll just be a small number of data sets, one of which will be missing. But we hope to kind of grow that over time to make it easier for researchers around the world who have their own autism data sets to be able to share it with people around the world. And the more data shared, the more biological insights that we can get, and just the more exciting research can be done. So one of the kind of really kind of important things I think about the Autism Sharing Initiative is that we have individuals involved with that project called AREPs. So AREPs are autistic individuals who provide input into the project. And sort of with increasing emphasis in many different research settings on like equity, diversity, and inclusion, we think that's really important. Making sure that the voices of those who are most directly affected by the research, which are autistic people themselves, that their viewpoints are represented in the design of the studies and how the findings are disseminated and stuff like that.
No, that's amazing. That's really exciting, and I'm sure it's a really important thing for those people as well. So that's amazing that they're being involved so heavily from the start of the project to the end of it, and then afterwards as well. So that's great. In terms of the future, what does the future look like for this work, do you think? Where is it sort of leading to? And then in general, what do you think is exciting about genomics right now and for the future of genomics?
Yeah, so I think two things that are quite exciting, both now and in the future, I guess, about genomics. I guess the first one, the more immediate one, is that the cost of whole genome sequencing is dropping quite a bit. So it used to be that sequencing just one person's genome cost like tens of thousands of dollars. So it was impossible to actually sequence, large groups of people at scale. And more recently, the cost of the single human genome was maybe around $1,000 Canadian, which is a lot lower, but it's still it's still limiting in terms of the number of people that you can sequence, depending on how much funding you have. But the major sequencing companies like Illumina and PacBio are now releasing new sequencing machines that I know CGEn is going to acquire, which is amazing. That further reduce the cost of whole genome sequencing. So just a reduction in cost, say from $1,000 per genome to say $500 per genome, or maybe later on $300 per genome. I mean, that's going to greatly increase, like double or triple the number of people that you can sequence using the same number of research dollars. So this will enable larger studies to be done. They'll have greater power in your statistics, and it'll just greatly increase the quality of the science that's done and the number of individuals that can participate. Also, in terms of the future, I think long read sequencing is going to be a major player. So all of the data in missing is from short read sequencing. So this means you kind of sample about 150 base pairs of the genome at a time. And for most purposes, that short read sequencing data is very good. We can get most types of genetic variants from that. But there's certain regions of the genome, especially regions that are really repetitive, that short read sequencing just can't really tell you that much about. So this long read sequencing is kind of the solution to that. So instead of 150 base pairs at a time, you can get thousands of base pairs at a time. And sometimes you can get up to, say, 50,000. So getting this long read sequencing data can really tell you a lot more about the genome. But the problem with it now is that it's a lot more expensive than short read sequencing. So long read sequencing is typically only done for a small number of samples that maybe you're really interested in. The other advantage to long read sequencing is that it also gives you information about methylation. So this goes back to the epigenetic studies that I was mentioning earlier, which is something that we didn't really look at in our missing paper. So with short read data, you need to do a different experiment in order to get information about methylation. But with the long read sequencing, especially with the newer technologies, it just sort of comes with this methylation information for free. It's sort of like a bonus. So not only do you get more informative reads that tell you more about these kind of repetitive regions of the genome, but you also get information about methylation without having to do another experiment, which is really valuable. So I think the only disadvantage is long read sequencing is currently a lot more expensive than short read sequencing, but it too is coming down in price a lot. And the instruments, the long read sequencing instruments that are being introduced now or will be available in the near future should be a lot cheaper than the previous ones. So it might make long read sequencing actually feasible for large cohorts of individuals, which I think is really, really exciting. I mean, that's going to be, I think, the holy grail in terms of being able to, detect almost everything about the genome in a single experiment.
And what does the time frame look like for that, do you think?
So I think there are long read sequencing machines that are supposed to be released in next year that will be about $1,000 per genome. I'm not totally sure if that pricing is sort of set in stone yet, but it should be somewhere in that ballpark from what I've heard. So that'll be the same price as what we paid for a short read genome just 2 or 3 years ago, which is, I think, very exciting.
Yeah, that's great. Thank you for sharing that. Is there anything else that you wanted to share about your work? It's been really great to hear all about it. Any last thoughts?
I think like aside from the technology, that just going back to what I was mentioning earlier about, how our understanding of the genome is still quite immature, but I think that is slowly being chipped away at. So the more experiments that people do to try to understand the genome, and this could be, experiments that are directly addressing a disease or just sort of more basic science experiments that just get at how does the genome work in a general sense. Every time one of these experiments is done or a paper on this is published, we learn just a little bit more about how the genome works. And then one of the things I think that's really valuable about doing genome sequencing is that, if we sequence someone's genome now, we may be able to figure out certain things, especially if they have a disease, but sometimes we won't. But as we learn more and more about the genome, we can go back to these previous genomes that have been sequenced and reanalyze them to try to figure out if there's anything that we missed before, because before we had way less knowledge about how the genome works, but now we have a lot more and we can actually maybe make sense of it a lot better than we could before. So these genomes that we're sequencing now are not just valuable to analyze now, but they'll be just super valuable as a resource in the future for many years, because we can continue to get more value out of them as we understand more and more about the genome.
And I guess that's where the role of like databases plays, right? In terms of being able to revisit the data and being able to reanalyze it.
Yeah, that's exactly right. And it goes back again to that Autism Sharing Initiative project that I mentioned. So the more people and researchers around the world that these genomes are available to, the more likely it is that, someone will have this light bulb moment that they'll see something in that genome that explains something.
Oh, that's great. It's very, very exciting. And I really, excuse me, appreciate you taking the time to share all this really great work that you're doing. Very exciting future ahead, it sounds like. And we'll also share the link to the Cell publication as well with this video so people can check out the work and learn more about it. So thank you so much, Brett, for taking the time.
Okay, thank you so much for inviting me. It was a pleasure.
Thanks. And take care. Thank you.
Thank you. Bye.